Welcome to the deep dive. Today we're plunging into a truly incredible hidden world, the one inside your own body, focusing on the machinery that's essential for, well, the continuation of life itself.
It really is amazing when you think about it, how all these tiny microscopic parts, cells, hormones, all work together in this incredibly complex way. It's fundamental biology.
Exactly. And we're going deep into Chapter 22 of Histology, a text in Atlas, the 8th edition, focusing entirely on the male reproductive system.
Right. Our goal here is to kind of unpack the microscopic anatomy, the cells, the tissues involved.
And not just list them, but connect them to how things work, why it matters clinically, and pull out some, surprising details along the way.
Yeah.
The very start of development right through to the journey of a single sperm cell.
It's that connection between the micro and the macro, the structure and the function that's so fascinating and clinically relevant, too.
Okay, so let's start big picture. What are the main parts we're talking about here?
Well, you've got the primary organs, the testes, of course. Then there's a whole network of tubes, the genital excurrent ducts.
Like plumbing, almost?
Kind of, yeah. And then the accessory glands, seminal vesicles, the prostate, bulbourethral glands, which add fluids, and finally the external parts, the penis and scrotum.
Got it. So the tests are central. What are their main jobs?
Two key functions. First, spermatogenesis. That's the process of actually making sperm, the male gametes.
Okay, making the cells.
Right. And second, steroidogenesis. That means making steroid hormones, chiefly testosterone.
And testosterone, that's huge, right? It does more than just, adult male stuff.
Oh, absolutely. It's crucial way earlier.
Yeah.
It directs embryonic development, basically telling the fetus to develop male characteristics.
Yeah.
It's fundamental for sexual dimorphism, the physical differences between males and females.
Wow. And all this cell production, it relies on cell division, right? Mitosis and meiosis.
Exactly. You have mitosis for just, making more identical cells for growth and maintaining the population. But then you have meiosis, which is this really specialized division process.
That's the one that has the chromosomes.
Precisely. It's essential to create those haploid gametes, the sperm, each with a unique genetic mix. We'll definitely get more into this.
Okay, so let's focus on the tests themselves. They're outside the main body cavity, in the scrotum. Why is that?
Location, location, location. It's critical. Spermatogenesis, making sperm, it needs a temperature that's actually lower than the core body temperature.
Lower. How much lower?
About 2 to 3 degrees Celsius lower, so roughly 4 to 5 degrees Fahrenheit.
That's quite a difference. Here's where it gets really interesting.
Yeah.
The body's design for temperature regulation here is quite ingenious.
It absolutely is, which raises the question, right? How does the body maintain that specific cooler temperature?
Yeah, how?
Well, it uses a couple of neat tricks. One is the pampiniform venous plexus. It's a network of veins wrapped around the testicular artery. It acts like a countercurrent heat exchanger. The cooler venous blood returning from the testes cools down the warmer arterial blood heading towards it.
Like a biological radiator.
Exactly. Plus, you have muscles, the cremaster muscle and the dartos muscle in the scrotal wall. They contract or relax depending on the outside temperature, pulling the testes closer to the body for warmth or letting them hang lower to cool off.
A built-in thermostat. Amazing. But let's rewind even further. How does the body even decide to make testes in the first place? Sex determination.
Right. So genetic sex, whether you have XX or XY chromosomes, is set the moment fertilization happens.
Okay.
But the gonads, the organs themselves, don't actually commit to being tests or ovaries until about the 7th week of embryonic development.
So there's a lag. What flips the switch for testes?
It's a specific gene on the Y chromosome called the SRY gene. SRY, it stands for sex-determining region Y.
The master switch.
Basically, yes. It encodes a protein called testis-determining factor, or TDF. And TDF kicks off this whole cascade, activating other genes like SOX9, SF1, and others, all leading down the path to developing tests.
So it's not just having a Y chromosome, it's that specific SRY gene doing its job. What if it doesn't work right?
That can happen. Mutations in the SRY gene can lead to something called Swyer syndrome. You have an individual who is genetically XY, but because the SRY switch didn't work, they develop externally as female. They don't have functional gonads, so they typically need hormone therapy later. It really highlights how critical that one gene is.
Really shows the power of a single gene. Okay, so the SRY gene flips the switch, testes start developing. What about hormones early on?
Good question. Early on, specific cells appear in the developing testes. Leydig cells start making testosterone, which is vital for developing the male internal duct system.
Okay.
And another type, Sertoli cells, produce something called Müllerian inhibiting factor, or MIF, sometimes called AMH, anti-Müllerian hormone.
And what does that do?
It actively causes the structures that would normally become the female reproductive organs, the uterus, fallopian tubes, to regress and disappear.
So it's actively suppressing the female path while testosterone promotes the male one.
Exactly. And then another hormone, dihydrotestosterone, or DHT, which is actually made from testosterone, is needed for the development of the external male genitalia, like the penis and scrotum.
A coordinated hormonal effort. And the tests themselves, they don't stay up inside the abdomen, do they?
No, they make quite a journey. They descend down into the scrotum, usually around the 26th week of gestation. It's guided partly by hormones like testosterone and INSL3, another hormone from Leydig cells, and also by a ligament called the gubernaculum.
What happens if they don't make it all the way down?
That's a condition called cryptorchidism, meaning hidden testis. It's actually quite common, especially in premature infants, but it affects maybe 1% of full-term newborns too.
Is that a problem?
It can be, yes. If the testes remain undescended, the higher temperature inside the body can cause irreversible damage to the sperm-producing tubules over time. It also significantly increases the risk of developing testicular cancer later in life.
Ah, okay, so it needs treatment.
Generally, yes. If they don't descend on their own within the first year or so, a surgical procedure called orchiopexy is usually done to bring the testes down into the scrotum and fix it there.
Okay, makes sense. So, assuming they descend properly, let's look inside a testis. What's the internal structure like?
Well, it's covered by this thick, dense connective tissue layer called the tunica albuginea. Think of it as a tough outer capsule.
Okay.
This capsule sends partitions, or septa, inwards, dividing the testes into maybe 250 or so little compartments called lobules.
Like segments in an orange?
Sort of, yeah.
Yeah.
And within each lobule, you find the real business end. 1 to 4 highly coiled tubes. These are the seminiferous tubules.
That's where the sperm are actually made.
Yeah, exactly. And packed in the connective tissue between these tubules are the Leydig cells, those hormone factories we mentioned earlier.
Got it. Tubules for sperm, Leydig cells for hormones. Let's dive into those seminiferous tubules. What's the lining like?
It's a very specialized, complex, stratified epithelium. Basically, 2 main cell types live there.
Okay.
First, you have the Sertoli cells. These are tall columnar cells that kind of form the structural framework of the tubule. They don't divide after puberty. Think of them as the support cells, or nurse cells.
Supporting the other cells.
Right. The other main population is the spermatogenic cells. These are the cells that are actually undergoing the process of becoming sperm. They're arranged in layers, with the least mature ones, the spermatogonia, down near the base, and the most mature ones, the spermatids, up near the central opening, the lumen.
So the Sertoli cells are like the scaffolding and the nurses for the developing sperm cells. What's immediately outside the tubule itself?
Surrounding the seminiferous epithelium is a layer called the tunica propria. It contains several layers of specialized cells called myoid cells.
Myoid. Like muscle.
Yeah, they have some contractile properties. They generate these slow, rhythmic contractions like gentle peristaltic waves that help to squeeze sperm and fluid along the tubule towards the exit.
Huh. Interesting. Another layer of complexity for moving things along.
Yeah.
Okay, let's jump back to those Leydig cells in the connective tissue between the tubules. The hormone factories.
Right. Leydig cells, or interstitial cells, they're usually large polygonal cells, often appearing pinkish, eosinophilic, and standard stains. They frequently contain lipid droplets, which are stores for making steroid hormones.
And you mentioned something curious about them earlier.
Yes, sometimes they contain these distinctive crystal-like structures called Reinke crystals. They're rod-shaped, and honestly, their function is still unknown. Probably protein products, but it's one of those little histological mysteries.
Always more to learn. But we know their main job is hormone synthesis.
Absolutely. Their cytoplasm is packed with smooth endoplasmic reticulum and mitochondria with special internal folds, classic features of cells that make steroid hormones, specifically testosterone.
And they're important throughout life.
Definitely. They're active in the fetus, making testosterone for that initial male development and INSL3 for testis descent. Then they quiet down a bit, but become very active again at puberty, driving sperm production and the development of secondary sexual characteristics. And in adulthood, they maintain spermatogenesis and male features. They even secrete a little oxytocin, which might help stimulate those myoid cells we mentioned.
And clinically, any issues with Leydig cells?
You can get Leydig cell tumors. They're usually benign, thankfully. But because they produce hormones, they can have effects. In young boys, they might cause precocious puberty. In adult men, sometimes they can actually produce estrogens, leading to effects like breast enlargement, gynecomastia.
Wow, hormones are powerful. Okay, let's get to the heart of the matter. Spermatogenesis, the actual process of making sperm.
Right. This is the entire sequence of events where the basic stem cells, the spermatogonia, develop into mature spermatozoa. It starts just before puberty, is kicked off by hormones from the pituitary gland, and then continues pretty much throughout adult life.
And it happens in phases.
Yes, it's helpful to think of it in three main phases. Phase 1 is the spermatogonial phase.
Okay, what happens here?
This involves the spermatogonia, the stem cells located right at the base of the seminiferous epithelium. They divide by mitosis. Some divisions produce more stem cells, ensuring a continuous supply. These are called type A spermatogonia.
So they renew themselves.
Exactly. And other divisions produce cells, type B spermatogonia, that are now committed to becoming sperm. A really key feature here is that when these cells divide, the daughter cells often remain connected by thin cytoplasmic bridges.
They stay linked together. Why?
It's thought to be crucial for ensuring that cohorts or groups of developing sperm cells develop synchronously at the same rate. They can share signals and molecules through these bridges. It's quite unique.
Fascinating.
Mm.
Okay, phase 1 is mitotic division and commitment. What's phase 2?
Phase 2 is the spermatocyte phase. This is where meiosis happens. The type B spermatogonia enter this phase, grow larger, and become primary spermatocytes.
Primary spermatocytes.
Yeah.
And they undergo meiosis.
Yes, 2 rounds of it. First is meiosis the first. This is a very long process, can take over 20 days in humans. During meiosis the first, the cell duplicates its DNA, homologous chromosomes pair up, and crucially, crossing over occurs.
When they swap bits of genetic material.
Exactly. And then the homologous chromosomes separate randomly. These two events, crossing over and random sorting, are massive sources of genetic variation. Meiosis site produces 2 secondary spermatocytes. These are now haploid in terms of chromosome number, but each chromosome still has 2 copies of DNA.
Okay, so genetic shuffling happens here.
Big time. Then comes meiosis the second, which is much quicker. The secondary spermatocytes divide again, this time separating the 2 copies of DNA in each chromosome. The result is 4 haploid spermatids. Each has half the number of chromosomes and only 1 copy of DNA per chromosome.
So what does this all mean? It's about genetic shuffling and specialization, all to create a unique gamete. Every single sperm ends up genetically different.
Precisely. You start with a diploid cell and end with unique haploid cells ready for the next phase.
Which is phase 3, the spermatid phase, also called spermiogenesis.
Right. This isn't cell division. It's a dramatic transformation, a remodeling process.
Right.
The round spermatids undergo massive changes to become the streamlined spermatozoa we typically picture.
Like a tadpole transforming into a frog, but on a cellular level.
That's a good analogy. It happens in stages. First, the Golgi phase. Little vesicles from the Golgi apparatus fuse to form a larger vesicle, the acrosomal vesicle, near the nucleus.
The acrosome, that's the cap on the sperm head, right?
Yes. In the next phase, the cap phase, this vesicle spreads out over the nucleus, forming the acrosomal cap. Meanwhile, the DNA inside the nucleus condenses incredibly tightly. Histones, the usual DNA packaging proteins, are replaced by smaller proteins called protamines. This makes the nucleus much smaller and protects the DNA.
Super condensed genetic package.
Extremely. Then comes the acrosome phase. The cell reorients itself, the tail, the flagellum, starts growing out, and mitochondria gather around the base of the flagellum to form the middle piece, the powerhouse for swimming.
Got it. Head, cap, condensed nucleus, growing tail, mitochondria for energy.
And finally, the maturation phase. Most of the excess cytoplasm is shed and phagocytosed, eaten up by the neighboring Sertoli cells. These shed bits are called residual bodies. What's left is the lean, mean, mature spermatozoon.
And the very last step, getting released.
Yes, that's spermiation. It's the final release of these elongated spermatids, now called spermatozoa, from the embrace of the Sertoli cells into the lumen, the central channel of the seminiferous tubule. This release is actually an active process, involving enzymes breaking cell connections.
Wow, that whole process is incredibly detailed. So let's look at the final product, the mature sperm cell.
It's a highly specialized cell. You've got the head, which is flattened and contains that supercondensed nucleus, and the acrosomal cap. That cap is full of hydrolytic enzymes like hyaluronidase and acrosin, needed to penetrate the layers surrounding the egg during fertilization. That's the acrosome reaction.
It's a key to getting in.
Exactly. Then there's a short neck, followed by the middle piece, which is packed with those mitochondria arranged in a spiral, providing the ATP energy for movement. Then the principal piece, which is the longest part of the tail, and finally a short end piece.
An incredibly optimized structure. But are they ready to swim and fertilize right after spermiation?
Not quite. They're morphologically mature, but not yet motile or capable of fertilization. They get pushed along by fluid flow and those myoid cell contractions through the seminiferous tubules into tiny straight tubules, then into a network called the rete testis, and then out of the testis via the efferent ductules into the epididymis.
Ah, the epididymis. We mentioned it earlier. That's where they finish their training.
You could say that. It's a very long, coiled tube, like 4 to 6 meters long if you uncoiled it. As sperm travel through the epididymis, which takes about 12 days, they undergo final maturation steps. They acquire the ability to move progressively forward, motility.
They learn to swim.
Pretty much. And other changes happen too, like further condensation of the DNA, shedding a bit more cytoplasm, and important modifications to their plasma membrane. One key change is called decapacitation. They gain factors on their surface that actually prevent them from fertilizing an egg too early. This needs to be reversed later in the female tract.
So the epididymis is crucial for both maturation and storage. Now, this whole spermatogenesis thing, it takes time, right? You mentioned the phases.
Yes, the timing is remarkably precise. In humans, from the initial division of a spermatogonium all the way through spermiogenesis takes about 74 days.
74 days.
Plus another roughly 12 days to transit through the epididymis. So you're looking at a total of about 86 days, almost 3 months, from the start of the process until a sperm is potentially ready for ejaculation.
That's why things like fevers or certain drugs might not show effects on sperm count or quality for a couple of months.
Exactly. The cells affected need time to complete their journey. And despite this long process, the output is massive. Around 300 million sperm are produced every single day in a healthy adult male.
300 million daily. That number is just staggering. Let's go back to those Sertoli cells again. We call them nurse cells, but they do way more.
They really are the workhorses of the seminiferous tubule. They provide physical support, nutrients, they mediate the effects of hormones like FSH and testosterone on the developing germ cells, they phagocytose those residual bodies and any degenerating germ cells.
The cleaners too?
Yep. And critically, they form the blood-testis barrier. This is formed by specialized tight junctions between adjacent Sertoli cells near the base of the epithelium.
Okay, a barrier. Why is that so important?
Think about it. The spermatocytes, spermatids, and spermatids, the cells undergoing and resulting from meiosis, are genetically different from the rest of the man's body cells. They have only half the chromosomes, and they express proteins that appear after immune tolerance is established early in life.
So the immune system would see them as foreign.
Potentially, yes. The blood-testis barrier divides the seminiferous epithelium into a basal compartment, where the spermatogonia are, and an luminal compartment, where meiosis and spermiogenesis happen. This barrier isolates the more mature, potentially antigenic germ cells in the luminal compartment from the immune system circulating in the blood and interstitial fluid.
It's like immunological privilege for the developing sperm. What happens if that barrier breaks down?
Breakdown of the barrier, maybe due to infection, trauma, or even sometimes after a vasectomy, can expose the immune system to sperm antigens. This can lead to the formation of anti-sperm antibodies.
And those antibodies can cause infertility.
They can. They might cause sperm to clump together, agglutinate, or impair their motility or ability to fertilize.
Wow. So the barrier is essential for fertility. Do Sertoli cells also secrete things?
Yes, they have both exocrine and endocrine functions. They secrete fluid into the lumen to help transport sperm. They secrete androgen-binding protein, ABP, which binds testosterone and keeps levels extremely high within the tubule, much higher than in the blood, which is essential for spermatogenesis.
Concentrating the key hormone right where it's needed.
Precisely. And they also secrete inhibin, a hormone that feeds back to the pituitary gland to specifically regulate the release of FSH, follicle-stimulating hormone, one of the key drivers of spermatogenesis.
A complex regulatory system. Okay, we've made sperm, they've matured a bit. Now let's follow their path out through the duct system and meet the glands that add the fluids.
Right, so from the seminiferous tubules, sperm move into the rete testis network, then through the efferent ductules, out of the testis itself.
Efferent ductules, what are they like?
They're short tubes connecting the rete testes to the epididymis. Microscopically, they have a characteristic scalloped or weighty-looking lumen because their lining has patches of tall cells with cilia, which help move sperm, alternating with shorter cells without cilia, which absorb most of the fluid.
So they concentrate the sperm by removing fluid.
Primarily, yes. Then it's into the epididymis, that long coiled tube we talked about for maturation and storage. Its lining is pseudostratified columnar epithelium, and the main cells, principal cells, have very long branched microvilli called stereocilia.
Stereocilia, not like true cilia that beat.
No, they don't beat. They're absorptive and secretory, involved in creating the right microenvironment for sperm maturation. The epididymis also has a progressively thickening coat of smooth muscle to help propel sperm during ejaculation.
And after the epididymis comes the
The
diaphragm.
ductus deferens, or vas deferens, this is a direct continuation of the epididymis traveling up through the spermatic cord into the pelvic cavity.
And it's known for being quite muscular.
Very. It has a remarkably thick wall composed of 3 distinct layers of smooth muscle, inner longitudinal, middle circular, outer longitudinal. This thick, muscular wall gives it a small, often star-shaped or folded lumen in cross-section and allows for powerful contractions during ejaculation to propel sperm quickly.
Which brings us to vasectomy again. That's where this tube is cut.
Exactly. A vasectomy involves cutting or blocking the ductus deferens, usually in the part that's easily accessible in the scrotum. It prevents sperm from traveling from the epididymis further along the tract. It's highly effective permanent contraception. Sperm production continues, but the sperm are just broken down and reabsorbed locally.
Okay. The ductus deferens then widens near the end.
Yes, it forms an enlargement called the ampulla, which also has some secretory function before it joins the duct of the seminal vesicle to form the ejaculatory duct, which passes through the prostate.
All right, perfect segue to the accessory sex glands. They add the fluid part of semen. Let's start with the seminal vesicles.
These are a pair of highly coiled, pouch-like glands located behind the bladder. They look really complex and folded internally.
And what do they contribute?
They produce the largest portion of the seminal fluid, maybe 65–75%. It's a thick, yellowish, alkaline fluid that's rich in fructose.
Fructose. Like fruit sugar.
Yeah, it's the primary energy source for sperm metabolism after ejaculation. Their fluid also contains other things like prostaglandins, clotting factors, and pigments that make semen fluoresce under UV light.
Prostaglandins. Aren't those involved in inflammation?
They have many roles. Here, they're thought to potentially influence muscle contractions in the female reproductive tract, maybe helping sperm transport. And despite the name, they're mostly made in the seminal vesicles, not the prostate.
Interesting. Okay, next gland. The prostate. The big one.
The prostate is the largest accessory sex gland, about the size of a walnut, located just below the bladder, surrounding the first part of the urethra, the prostatic urethra.
And its main job.
It secretes a thinner, milky, slightly alkaline fluid that contributes about 25–30% of the semen volume. This fluid contains citrate, another nutrient for sperm, enzymes like prostate-specific antigen, PSA, prostatic acid phosphatase, PAP, and fibrinolysin.
PSA is the one used in screening, right?
Yes. PSA is an enzyme that helps liquefy the semen coagulum shortly after ejaculation, allowing sperm to swim freely. It's also measured in blood tests as a marker for prostate conditions, though, as we discussed, its use for universal cancer screening is complex.
And the prostate has different zones. Why is that important?
Yes, anatomically and clinically, it's divided into zones. The peripheral zone is the largest part, at the back, and it's where most prostate cancers arise. The central zone surrounds the ejaculatory ducts. The transitional zone and periurethral zone surround the urethra more closely.
And those relate to BPH.
Exactly. Benign prostatic hyperplasia, BPH, the common non-cancerous enlargement of the prostate in older men that causes urinary problems, typically originates in the transitional and periurethral zones right around the urethra. It's often driven by DHT, that potent testosterone derivative.
So BPH usually affects urination because of where it grows, while cancer often starts further back.
Generally, yes. That's why digital rectal exams can sometimes detect cancers in the peripheral zone. Treatment for BPH might involve drugs to relax smooth muscle or shrink the gland, while prostate cancer treatment depends on how advanced it is and might involve surgery, radiation, or hormone therapy to block testosterone.
Complex gland with major clinical significance.
Okay, one more pair of glands.
The bulbourethral glands, also called Cowper's glands, these are small pea-sized glands located near the base of the penis.
And their contribution.
They secrete a clear, slippery, mucus-like fluid during sexual arousal before ejaculation. This pre-ejaculate helps to lubricate the urethra and neutralize any acidic urine residue, making the path more hospitable for the sperm that follow. Preparing the way. So putting it all together, semen.
Semen is the final mixture. It's sperm from the testes, via the epididymis and ductus deferens, plus the secretions from the seminal vesicles, the prostate, and the bulbourethral glands.
And overall properties.
It's typically whitish-gray, slightly alkaline, pH around 7.7, to counteract the acidity of the male urethra and the female vagina. Volume is usually 2 to 5 milliliters per ejaculate, containing anywhere from 20 million to over 100 million sperm per milliliter. The seminal vesicle fluid provides volume and fructose. The prostate fluid adds enzymes and alkalinity.
Okay. Now, finally, the delivery system, the penis. Structurally, what allows it to function?
The main body of the penis is made of 3 columns of erectile tissue. There are 2 corpora cavernosa side by side on the dorsal side top, and 1 corpus spongiosum underneath, which surrounds the urethra. All 3 are encased in that tough connective tissue layer, the tunica albuginea.
And erectile tissue means it can fill with blood.
Exactly. These tissues are like sponges filled with interconnected vascular spaces. Erection is fundamentally a vascular event.
How does it work?
It's initiated by signals from the central nervous system, often via parasympathetic nerves. These signals cause the release of neurotransmitters like acetylcholine and, crucially, nitric oxide, NO, at the smooth muscle cells in the walls of the small arteries, helicine arteries, that feed the erectile tissue.
Nitric oxide, okay.
NO triggers a chemical cascade inside the smooth muscle cells, leading to an increase in cyclic GMP, cGMP. This causes the smooth muscle to relax.
Relaxation leads to erection. That seems counterintuitive.
It does, but think of it this way. When the smooth muscle in the artery walls relaxes, the arteries dilate widely, allowing a rush of blood into those vascular spaces in the corpora cavernosa. As these spaces fill rapidly, they expand and compress the small veins that normally drain the blood away, trapping the blood inside. This engorgement causes the penis to become rigid and erect.
Relaxation lets blood in, the pressure traps it. So what happens in erectile dysfunction, or ED?
Yes. ED is the consistent inability to achieve or maintain an erection sufficient for intercourse. It can have many causes: vascular problems, nerve issues, hormonal imbalances, psychological factors, medication side effects.
And treatments often target that NO pathway.
Yes, drugs like sildenafil, Viagra, and similar medications are phosphodiesterase type 5, PDE5 inhibitors. PDE5 is the enzyme that normally breaks down cGMP. By inhibiting this enzyme, these drugs help maintain higher levels of cGMP, prolonging the smooth muscle relaxation initiated by NO and facilitating erection.
But they need that initial NO signal to work, right?
Absolutely critical point. These drugs enhance the natural pathway, they don't initiate it. So if there's significant nerve damage, say from radical prostate surgery or severe diabetes that prevents the release of acetylcholine and NO in the first place, these pills won't be effective. It really highlights the precise interplay of nerves, blood vessels, and molecular signals.
A perfect example of structure, function, and clinical application. And the final step is ejaculation.
Right. Ejaculation is primarily driven by the sympathetic nervous system. It causes rhythmic contractions of the smooth muscle and the epididymis, ductus deferens, seminal vesicles, and prostate, forcefully expelling the semen out through the urethra.
What an incredible journey we've taken. Seriously, from looking at that SRY gene kicking things off, through the mind-boggling details of spermatogenesis, meiosis, spermiogenesis,
The cellular remodeling.
all the way through the duct systems, the glands adding their contributions, the vascular events of erection. It's just astounding.
It really is.
So what does this all mean? I mean, it's a testament to the unbelievable precision and interconnectedness within our own bodies. This system is constantly working, constantly renewing itself, preparing for the possibility of creating the next generation.
And if we connect this to the bigger picture, it really underscores how these fundamental biological processes, right down to the molecules and the specific cell junctions like the blood-testis barrier, are so exquisitely regulated. And it shows how disruptions at almost any point in that chain, genetic, hormonal, structural, vascular, can have really significant clinical consequences affecting fertility or overall health. It really makes her wonder, doesn't it? How many other functions in our body that seem simple on the surface are actually built on this kind of incredible, intricate, and frankly quite delicate foundation?
That is a great question to ponder. The sheer scale of production, 300 million sperm a day. The complexity, the tight regulation, the protective barriers. It really drives home the resilience, but also the potential vulnerability of these biological systems. And understanding it at this level is key for developing better treatments, isn't it?
Absolutely. From infertility therapies to cancer treatments to managing hormonal issues, that detailed histological and molecular knowledge is the bedrock.
We hope this deep dive is giving you, our listener, a new appreciation for this amazing microscopic world. There's so much complexity constantly at work within us. Keep exploring, keep questioning. Until next time on The Deep Dive.